90%. That single number destroyed a story that the entire world had been told for 500 years. Not 90% Spanish, not 90% mixed, 90% indigenous. Through the maternal line, through the blood of mothers and grandmothers going back thousands of years, the Mexican people were never a balanced blend. They were something far older, far deeper, and far more extraordinary than any textbook ever admitted. And when modern genetic science finally turned its full power onto the DNA of 130 million people, what came back was not a confirmation of the official story. It was a demolition of it, complete, permanent, and without a single apology. This is not a story about politics. It is not about borders, flags, or language. This is a story about what is written inside the cells of human beings at the most fundamental biological level possible. And that story, once you actually read it, the way scientists have now learned to read it, changes everything you thought you knew about one of the most important populations on the planet. Let us start with the lie. Not a malicious lie, but a comfortable one. The kind of lie that gets repeated so many times, it stops feeling like a lie and starts feeling like common sense. The story told about Mexico went like this. Spanish conquerors arrived. They mixed with indigenous people, and the modern Mexican was born. A blend, half and half. European and indigenous in rough balance with a small African element added in, the mestizo, a product of three rivers converging into one national identity. Clean, simple, manageable. That was the story in the schools, in the history books, in the political speeches. It was also, as genetic science has now definitively proven, deeply wrong. When researchers analyzed mitochondrial DNA, the genetic material that passes exclusively from mother to child, generation after generation without mixing or dilution, the data from over 2,000 samples collected from across every region of Mexico told a completely different story. Nearly 90% of all maternal lineages in the modern Mexican population belong to four ancient indigenous haplogroups, A2, B2, C1, and D1. These are not European haplogroups. These are not African haplogroups. These are the genetic markers of the people who were already living on the American continent for thousands of years before any European ship ever crossed the Atlantic. Through the mother's line, the Mexican people are overwhelmingly, undeniably ancient indigenous American. That is not an opinion. That is the molecular evidence extracted from human cells. But here is where the story becomes more complicated and far more disturbing. Because DNA tells multiple stories simultaneously, and those stories do not always agree with each other. The maternal line gives you one history. The paternal line gives you another. And in Mexico, those two histories are so different from one another that they almost seem to belong to separate worlds. Because in a very real sense, they do. When scientists examined the Y chromosome, which passes exclusively from father to son, the picture was almost the direct opposite of what the maternal line revealed. Among admixed Mexicans, paternal ancestry came back as approximately 65% European, around 31% indigenous, and roughly 4% African. So through the fathers, the genetic story of Mexico is predominantly European. Through the mothers, it is predominantly indigenous. The same body, the same person, the same Mexican family carries two completely separate ancestral histories in two completely separate parts of the genome. One inherited from the conquerors, one inherited from the conquered. Scientists do not describe this simply as mixing. The technical term is asymmetric admixture, and the reason it is asymmetric is not biological. It is historical. It is the direct genetic fingerprint of what actually happened during the colonial period. European men came in enormous numbers. Indigenous women survived in enormous numbers. The population collapse that followed the arrival of European disease was so catastrophic that it restructured the very architecture of the human genome in Mexico. The DNA preserved what the history books minimized. European men contributed their paternal genetics into the population on a massive scale. Indigenous women provided the maternal foundation on which the entire modern Mexican population was built. The result is a people whose cellular blueprint is split, carrying the biology of power through one channel and the biology of survival through another. And that brings us to the part of this story that almost nobody talks about: how far back the real origin of the Mexican people actually goes. Because you cannot understand what Mexican DNA means without going back not 500 years, but 15,000 years. You have to go back to a frozen land bridge, to a continent that no human being had ever stepped foot on, to a moment in prehistory when a group of people walked out of northeastern Asia and into an empty world that would eventually become the Americas. The genetic evidence is absolutely clear on this point. Indigenous Mexican DNA carries unmistakable connections to populations from North and Central Asia. The haplogroups found in the mitochondrial DNA of modern indigenous Mexicans are the same haplogroups found in populations from Siberia and populations from indigenous communities in Canada. This is not coincidence. This is the preserved genetic echo of the original migration event that populated the entire Western Hemisphere. The ancestors of the Maya, the Aztec, the Zaptec, the Mixtec, and every other civilization that rose and fell on Mexican soil came from Asia. They followed large animals across a land corridor connecting what is now Siberia to what is now Alaska during the last ice age, when sea levels were dramatically lower than today. They had no idea they were entering a new continent. They were surviving. And over thousands of years, their descendants spread southward and populated 2 entire continents, ultimately building some of the most sophisticated civilizations the ancient world ever produced. I-chromosome data from indigenous Mexican populations confirmed this directly, showing the southward settlement of the Americas through that land bridge and connecting native Mexicans biologically to peoples from south-central Siberia and northern Canada. The genetic fingerprints are not faint. They are clear enough that researchers can trace the route ancient populations took as they moved deeper into the Americas over millennia. Every indigenous Mexican alive today carries Asia inside their genome. They have always carried it. The proof has simply been sitting in their cells, waiting for science to assemble the tools capable of reading it. Here is what makes this scientifically disturbing. The Olmec left no recoverable maternal lineage and no recoverable paternal lineage in the genetic record. Modern genetic tools, which can detect population signals from thousands of years ago, cannot find a clear Olmec echo in the DNA of modern Mexican people. A civilization that built the foundations of all subsequent Mesoamerican culture has disappeared, not just from the historical record, but from the biological record. Scientists can point to where they lived. They can show you the colossal heads they carved from solid basalt, but they cannot tell you where these people biologically came from or where their genetic descendants are today. The deepest root of Mexican civilization is, in the language of genetics, a black hole. And that is not a minor gap. That is a foundational mystery that sits at the very core of Mexican genetic identity. Now, 1,600 years after the Olmec disappeared, something happened that the genetic record reveals with shocking clarity. In 2024, a landmark study published in the journal Nature described the results of sequencing data from nearly 150,000 adults enrolled in the Mexico City Prospective Study, one of the largest genetic studies ever conducted anywhere in the world outside of Europe. And what it found went far beyond ancestry percentages. It found something medically alarming. The indigenous segments of the Mexican genome carry specific patterns of genetic variation that are unlike those found in European or African segments of the same genomes. Specifically, those indigenous segments show lower levels of coding variation, but a significantly higher concentration of what scientists call homozygous loss-of-function variants. In plain language, genetic positions, where when a person inherits the same damaged variant from both their mother and their father, a gene simply stops working. These patterns are the biological consequence of thousands of years of relative geographic isolation. A population separated from the global gene pool long enough develops its own genetic profile, and when that profile meets the modern world, with its processed foods, environmental exposures, pharmaceutical drugs calibrated for other genetics, and disease patterns that have no pre-colonial precedent, the results are not random. They are predictable, they are measurable, and they are being systematically ignored. The Origin Cohort Study, published in early 2026 and enrolling over 83,000 individuals from 18 metropolitan areas across 19 Mexican states, confirmed an average admixture of 61.8% Native American, 32.4% European, and 5.1% African ancestry in its genomic analyses. But it also found something the numbers alone do not capture: a population carrying a 40% rate of obesity, elevated blood pressure levels, and elevated blood glucose averages. These are not coincidences. These are the interaction between a specific ancient genetic architecture and a modern environment that was never designed with that architecture in mind. The Mexican Biobank Project genotyped 6,057 individuals from 898 localities across all 32 Mexican states. And in 2026, its clinical findings were published in Nature Medicine. The results characterized over 42,000 clinically relevant genetic variants across the entire country and demonstrated something with profound consequences. The genetic risk scores, drug dosage guidelines, and diagnostic tools that power modern medicine around the world were built on reference panels derived almost entirely from European populations. They are being applied to a population whose genetic profile is fundamentally different. Every day, in clinics and hospitals across Mexico and across the United States, where 37 million people of Mexican descent now live, medical decisions are being made using tools that were not calibrated for the biology of the people they are supposed to serve. This is not a small technical error. This is a structural failure of the global medical system, and scientists have been documenting it with increasing urgency. The regional dimension of this story is just as important as the national average. Mexico is not genetically uniform. It never was, and the data makes clear it still is not. Indigenous populations in distinct regions of Mexico can be as genetically differentiated from one another as European populations are from East Asian populations. That is not a slight exaggeration for emphasis. That is the direct finding of researchers, who mapped genetic distance between indigenous Mexican groups. In the southern and southeastern states of Oaxaca and Chiapas, indigenous ancestry reaches between 70 and 90% of the total genetic composition of local populations. In the northern states, European ancestry can range from 50 to 60%. Within the borders of a single nation, you have populations whose genetic profiles differ as dramatically as populations from completely different parts of the world. They are all called Mexican. The single label conceals a biological diversity that is almost incomprehensible in its depth and complexity. Then there is the dimension of this story that has been suppressed most deliberately and most completely. The African genetic contribution to Mexico is not a footnote. Enslaved Africans were brought to Mexico in significant numbers during the colonial period, particularly to coastal regions of Veracruz and Guerrero, and the genetic signal of their presence did not disappear. In certain coastal communities in those states, African ancestry reaches between 6 and 9% of the total genetic profile. These are not traces. These are communities with living biological connections to West Africa, to the transatlantic slave trade, to a history that the Mexican state did not officially acknowledge in its census for generations and did not teach in its schools. The people of the third root, as they have sometimes been called, were invisible to the government while their DNA held the full record of who they were and where they came from. Genetic science restored what official history chose to erase. Now consider the immune system. At the MHC region, one of the most critical zones of the human immune genome, Mexican populations show a specific overrepresentation of African ancestral genetic variants compared to the rest of their genome. The selection pressure that produced this pattern is among the strongest ever measured in the human species. Researchers have compared it directly to the intensity of lactase selection in Northern Europeans and the sickle cell trait in African populations. What that means is that the immune system of the Mexican population has been shaped by evolutionary forces, unlike those experienced by any other large group of people on Earth. The specific combination of ancient indigenous American immunity, colonial era European exposure, African origin immune genes, and the unique disease environment of Mesoamerica over centuries has produced an immune architecture that is genuinely unprecedented in the human genetic record. And this unprecedented system is being assessed every day by diagnostic tools that do not know it exists. The most recent large-scale genomic work on Mexican populations, including studies published through 2025 and 2026, is beginning to assemble something that did not exist even 5 years ago: a comprehensive population-level genetic map of one of the world's most complex and most understudied peoples. The Mexican Biobank Project identified that polygenic risk scores derived from its own population-specific data consistently outperform scores derived from European datasets for multiple health traits in Mexican individuals. In plain terms, the genetic tools built from Mexican DNA work better for Mexicans than the tools that have been used on them for decades. The tools that were supposed to be universal were never universal at all. They were European tools applied globally under the assumption that European genetics could stand in for all of humanity. That assumption is now scientifically demolished. There's also a dimension of this story that points not backward to conquest and collapse, but forward to something remarkable about persistence. Researchers analyzing multiple generations of Mexican genetic data have noted something that directly contradicts the expectation that globalization and modernization would gradually dilute indigenous genetic identity into a generic global average. In many regions of Mexico, the indigenous genetic signal in recent population samples is actually stronger relative to other ancestry components than in older samples. The ancient DNA, the genetic inheritance of people who crossed a frozen land bridge from Asia 15,000 years ago and built pyramids that still stand, is not fading. It is holding. It is durable in a way that 500 years of colonial pressure, forced religious conversion, demographic collapse, and cultural erasure could not override. The genome preserved what history tried to destroy. And the ancient lineages of Mexico did not stay in Mexico. Mitochondrial DNA from pre-Aztec civilizations, including the Maya and the people of Teotihuacan, did not simply disappear when those civilizations fell. Those maternal lineages spread across the entire length of 2 continents. Indigenous peoples from the Arctic carry mitochondrial connections to ancient Mesoamerican populations. Indigenous communities in Chile and Argentina carry maternal lineage connections to the Maya. The people who built civilization in what is now Mexico were not a regional population. Their genetic legacy runs from the Arctic Circle to the southern tip of South America. In a biological sense that is measurable and documented, the ancient Mexicans were the ancestral heart of an entire hemisphere. And then the disease came. In the decades following 1519, and somewhere between 50 and 90% of the indigenous population of Mesoamerica died. Not in battle, though battles were fought. They died of smallpox, measles, typhus, and influenza, diseases against which they had no immunity because their isolated genetic history had never encountered those pathogens. Entire languages went silent. Entire genetic lineages that had existed for 10,000 years simply ended. The diversity built over 15 millennia of isolated evolution was compressed into a fraction of its former range within a few generations. The Mexico City Perspective Study's genomic data reveals this in the DNA itself through specific patterns of relatedness and runs of homozygosity that tell the story of a population catastrophically reduced and then rebuilt from a much smaller surviving pool. The biological evidence of what happened 5 centuries ago is literally visible in the genome of people alive right now. The genocide is not just historical record, it is living molecular code. In January 2026, Nature Medicine published findings from the Mexican Biobank documenting 42,769 clinically relevant variants across the Mexican population, accessible through a publicly available web application designed to make this data available to medical researchers working with Hispanic populations worldwide. The significance of this cannot be overstated. For the first time, there's a population-specific genetic reference built from actual Mexican genetic diversity. Not approximated from European data, not generalized from a handful of samples, but constructed from thousands of individuals representing all 32 states and hundreds of distinct localities. This is the foundation on which future medicine for this population will be built. It is the beginning of what should have existed decades ago. The 37 million people of Mexican descent living in the United States carry one of the most genetically complex profiles of any population group on the planet. They are part Siberian-descended Indigenous American, they are part Iberian European, they are part West African. They carry the distinct genetic signatures of dozens of pre-Columbian civilizations that were themselves as different from one another as populations from different continents. They do not fit in any single demographic box. No health risk model built on European genetics captures their actual risk accurately. No pharmacogenomic guideline calibrated on European drug responses reflects their actual responses reliably. Every policy, every research study, every clinical protocol that treats them as a homogeneous category is operating on a scientific error with real consequences for real people. What happens now is the question. The science is assembled. The data is increasingly available. The understanding of Mexican genetic complexity has reached a level of resolution that was impossible even 10 years ago. But the gap between what the science knows and what the world's medical infrastructure does about it is not closing nearly fast enough. Researchers have been clear in their published work. Recognizing Mexico's genetic diversity is essential for building more accurate, more inclusive, and more equitable biomedical science. The gap is not just a research problem. It costs lives in misdiagnosis. It costs lives in drugs that perform differently in this population than the trials predicted. It costs lives in risk models that tell people they are safe when their specific genetic profile says otherwise. The Mexican people were never who the world thought they were. They were never a simple blend of European and indigenous, conveniently mixed and neatly categorized. They were something that took 15,000 years of human migration, civilizational rise and fall, the catastrophe of colonial contact, the survival of an ancient genetic legacy through everything that tried to destroy it, and the painstaking labor of modern genomic science to begin to truly understand. Their DNA contains Siberia and Spain and West Africa, and the memory of pyramids and the echo of a population collapse that was arguably the largest demographic catastrophe in human history. All of that is still in the genome. All of it is still being read. And the most important chapter of what that means for medicine, for identity, for science, for the hundreds of millions of people whose health and lives depend on getting this right, that chapter is only now beginning to be written.